In this screencast video lecture, we are going to see about the structural organization, that is some 4 different level of organization of protein in the living systems. Proteins have a complex structure that is organized into 4 levels: primary, secondary, tertiary and quaternary levels. Each level of structure is crucial for the protein to function there in the living system and its ability to interact with the other molecules, say for example like substrate or with the DNA molecule during the replication. This interaction could be possible only with a particular level organization of the protein. Now we look at the details of the primary structure. The primary structure of protein is defined as a linear sequence of amino acids that have been present there in the polypeptide chain. It has been held mainly by the peptide bonds. Next, we look at the salient points of this primary structure. The primary structure depends upon the specific sequence of amino acids from the N terminal, that is amino end, to the C terminal, carboxyl end of the protein molecule. The sequence is encoded by the corresponding DNA, and any change in the DNA sequence that could be resulted by the mutation will be reflecting there or altering in the amino acid sequences. The primary structure dictates the protein's higher order structure, that is other secondary, tertiary structures are all dictated by the primary structure. Importance: the order of amino acids determine how the protein will fold into its secondary and tertiary structures, mainly affecting its functionality. Next one, changes in this primary structure of the protein that could be resulted by the point mutation may lead to causation of certain diseases. For example, a single amino acid change in the hemoglobin results in the formation of sickle cell anemia. Next, we look at the points related to the secondary structure. Secondary structure refers to localized folding patterns within a polypeptide chain that are in turn stabilized by the hydrogen bonds between the backbone atoms that have been present there in the protein molecule. Its salient feature is of two main types. One is the alpha helix and another one is a beta pleated sheet. The random coils are those that can able to connect the alpha helix and beta pleated sheet. Here in alpha helix, a coiled structure is stabilized by hydrogen bonds formed between every 4th amino acid. The next one is beta pleated sheet. It's a sheet-like structure wherein segments of the polypeptide chain run alongside each other, forming hydrogen bonds. The random coils are loops. They are non-repetitive regions that can able to connect the helixes as well as the beta pleated sheet. Now we look at the importance of the secondary structure. The secondary structures contribute to overall stability and flexibility of the protein. They in turn dictates the framework for the next level, that is tertiary level organization of the protein. Specific configuration of the secondary structures are important from the standpoints of the function of enzymes, even receptors that have been embedded in the membrane, and transport protein that are involved in the transport of various molecules. For example, the alpha helices in membrane proteins allow them to span into the entire lipid bilayers of the cell. Next one is a tertiary structure. The tertiary structure refers to the overall 3-dimensional folding of a single polypeptide chain. It has been in turn driven by the interaction between the side chains of the amino acid, that is between the R groups of the amino acids. Then the salient points of the tertiary structures include: they are stabilized by hydrophobic interaction that allows the non-polar amino acid side chains to cluster away from the water. Then hydrogen bonds mediates the arrangement between the polar side chains of the amino acids. Ionic bonds interaction facilitate the interaction between the positive and negatively charged side chains of the amino acid. Disulfide bridges refers to covalent bond that have been formed between the sulfur atom of the cysteine residues. This tertiary structure serves as an important one for each protein as it's critical for the functionality of the protein. Next, we look at the points related to importance of the tertiary structure. Tertiary structure helps in the functional domain formation. That is, many proteins will have a distinct region which are technically referred as the domains in the tertiary structure that are playing a major role in functioning of this protein. Example, there will be certain specific binding sites for the substrate that could able to catalyze the reactions. Second one, enzyme activity. The active site of an enzyme where substrate binding occurs is formed mainly by the tertiary structure, which I have narrated before also. Next one, the protein stability. Tertiary structure is the one which provides a stability there for the proteins to be functional under a varied environmental conditions, such as changes in the temperature or pH of the environment. The next one is the quaternary structure, or the final level hierarchical structure of the protein. It exists when protein consists of two or more polypeptide chains, that is subunits, which may be identical or different. These subunits are held together by a covalent interaction, basically of hydrogen bonds and hydrophobic interaction, and less commonly present interactions include the covalent bond formation between the disulfide bridges of the cysteine molecule. The salient points of this structure is many functional proteins such as a hemoglobin are made up of multiple polypeptide chains. So their proper functioning is dependent upon the quaternary structure of the protein. The subunits can be of the same type, that is of a homodimers or homotetramers, or they can be of a different types also, which is referred as a heterodimers or heterotetramers. Interaction between the subunits often influences the protein function, mainly through the cooperativity. Example for this is the binding of oxygen and carbon dioxide to a same hemoglobin molecule is achieved by principle of cooperativity. Next, we look at the importance of the quaternary structure. The first one is the regulatory function. That is, quaternary structure of a protein could regulate their activity in a much more fine way. This could be best understood by studying about certain allosteric proteins that can able to bind to a ligand at one subunit, whereas the other subunit activity could be affected. Such kind of proteins will be commonly having a standard quaternary structure that could help in a proper regulation there of the function. Next point is increased stability. The quaternary structure commonly provides a much increased stability for that particular protein, especially against the harsher environment. Say a high temperature or a low temperature could affect a protein's function, but that could be better guarded by the quaternary structures of the proteins. They can also able to resist the degradation. The next one is a cooperative binding. Here the classical example is a hemoglobin molecule, which exhibits the cooperative binding, wherein binding of one molecule to one subunit affects the binding affinity of the other subunits. Here the subunits of the hemoglobin have been involved there in binding carbon dioxide and oxygen, which has been technically referred as the cooperative binding of the molecules. And finally, we try to understand the importance of this protein structure, some overall functions. These protein structures offer a stability and proper folding there for the proteins, which is referred as the structural integrity of the proteins could be maintained. Then the protein structure offers some specificity functions for the proteins. I mean, it allows the proteins to interact with some specific molecules such as ligands or substrates, or even in certain cases such as a DNA. That is, the protein can able to interact with the DNA and they help in the replication of the DNA molecule or in the transcription of the DNA molecule. So, a high specificity of the protein is required there for performing a biological function inside the living cell. The next important point concerned with the overall structure of the protein is the diseases that are associated with the misfolding of the protein. That is, proper structure has not been obtained and a misfolding there is happening there in the protein. This leads to various diseases such as Alzheimer's disease, Parkinson's disease and prion-based disease, which are basically occur due to the misfolding or aggregation of the protein.